Good morning, everybody, and welcome to the Drug Hunter Flash Talk. I'm your host, Dennis Koster, and we're happy you're here today. We'll be talking about physics-based applications of modeling for medicinal chemists with Aleksey today. Welcome to the show, Aleksey. How are you doing this morning?
I'm doing great. Really excited to share how the physics-based modeling can help medicinal chemists and their decision making.
Awesome. Yeah, no, we very much look forward to that. Without further ado, let me introduce Aleksey to you. Aleksey Gerasyuto is a medicinal chemistry leader with extensive experience in computationally driven drug discovery and a champion for the predict first approach using modern physics-based methods and AIML methods as well. In his current role at Schrödinger, Aleksey is leading the medicinal chemistry team and co-leads the strategic discovery portfolio oversight of the therapeutics group. He has contributed to establishing and advancing several successful and productive collaborations, including the one with Morphic Therapeutics, Petra Pharma, BMS, and Novartis. Prior to joining Schrödinger, Aleksey held positions of increasing responsibility at PTC Therapeutics, which are one of the pioneers of RNA biology with targeting RNA with small molecules. And Aleksey has contributed to the discovery of several clinical candidates, including PTC 518 for Huntington's disease, which is currently in phase 2, Morph 057 for IBD, which is also in phase 2, SGR 3515 for solid tumors, and LOXO 783 for breast cancer and other tumors, currently in phase 1. Aleksey received his PhD from the University of Wisconsin in medicine and his bachelor's and master's degree in chemistry from the Higher Chemical College of the Russian Academy of Sciences. So welcome to the show, Aleksey. Really a pleasure to have you today.
Thank you so much, Dennis. Great to be here.
And yeah, in the good old tradition of the Drug Hunter Flash Talks, we start this with a little bit of icebreaker questions. So, Aleksey, can you tell us a little bit more about what inspired you to start a career in drug discovery?
Yeah, so that's an interesting question, Dennis. Actually, during my undergraduate and graduate research days pursuing the organic synthesis, I was 100% set on pursuing the academic career. Back then, I thought that's exactly where all the real science is actually happening. But as I was graduating, I had two industrial options available as an alternative to pursuing the academic career. And what got really me excited is the intriguing science by this very small biotech back then, PTC Therapeutics, which was going after targeting complex RNA biology with the small organic molecules. I really didn't fully understand all of the complexity of the biology, but I thought, well, if I would continue doing organic synthesis, why not do it also basically being the part of this journey, exploring this novel biology, pharmacology, and potentially treat the human disease. So I think it really distills down to the passion of integrating different disciplines, and in this case, it was bringing organic synthesis. I was really an expert in with exciting biology, and here I am pursuing the career in drug discovery.
Amazing. I mean, I think I had a similar similar way into the industry too. That's amazing. Can you tell us a little bit more about your favorite molecule and why that is your favorite molecule?
Yeah, that's the tough one. As I'm thinking about it, right? Picking one is just so difficult. There are so many great molecules, the industry comes up with. But I think the one molecule which really continues to fascinate me over the last several years is orforglipron, the oral GLP-1 agonist. It's just, fascinating, amazing molecule, in the beyond rule of five chemical space, yet very early bioavailable, have really low dose in the clinic. So it has very interesting, complex structure. And I really just can't wait to see the whole medicinal chemistry story, how it was actually discovered. So hopefully we can see that soon.
Yeah, absolutely. It has a wonderful acid bioisoster as well.
Yeah.
That's kind of non-obvious.
Yeah.
Love that structure as well.
Yeah.
Really
So
cool.
every basically substituent on that molecule was put there for a good reason. So yeah, I really can't wait to see the story, how it was discovered.
Totally, totally. So what are you most excited about today in drug discovery?
Yeah, so it's also a good question. I think what is really amazing nowadays, and I think this definitely changed over the last maybe a couple decades, is the speed with which the drug discovery advances are really being made. Nowadays, I sometimes feel actually completely overwhelmed keeping a pace with what's happening. So I think that's really exciting, right? Basically, every day you wake up to some really exciting news, and yes, I think that's exciting. And of course, I think the evolution of different predictive methods, right, which help us being more efficient in all stages of this drug discovery and development process. Early discovery, also some of the methods which are now being developed to help us in the clinical trials. And I wholeheartedly believe that we probably only have seen the small tip of the iceberg. I really believe that all of these methodologies which are now being developed will continue helping us being more and more efficient as these methods evolve of different kinds, right? Physics-based, AI, ML. I think this will be an interesting space. I really believe that this will continue to happen in drug discovery.
Yeah, absolutely. No, we're, I think, as you pointed out, at a very interesting point in drug discovery. And I'm like personally very happy to be alive at this time and be working in this super exciting field. Yeah,
Yeah.
Aleksey. So, I mean, you have so many compounds in the clinic, and we probably have a few really, young audience members here, maybe just starting their career in medicinal chemistry. What would you tell your younger self if you were to, start your career over again? What kind of advice would you give your younger self, and maybe the audience members here today that are just starting their careers?
Yeah, so I think the advice I would actually give to myself and anyone else taking on this career path is fearlessly challenge the stereotypes. I remember, learning the medicinal chemistry early on in my career, and all the things about, different rules of five, all the functional groups, which are absolute no-go for drug design. Then just to see many and many of those rules, constantly being broken and challenged as we continue advancing, science and pushing the boundaries. So I think, again, I was always a big fan of taking risks and still is. So I find this very rewarding from the standpoint to be in drug discovery and constantly changing this, stereotypes, taking risks, and then, identifying new and novel discoveries. So
Yeah, I couldn't agree more. I mean, you got to challenge the dogma, and those are the most interesting discoveries that you will make if you do. So
yeah.
I guess we'll learn more about one of those discoveries today or more. Maybe my last question. What's your favorite thing about Drug Hunter?
Yeah, so I don't think I will be the only one with this answer, but I'm a really big fan of your curated pattern search you introduced late, I think early last year. So I really love scrolling through the list every month and then looking for the targets, which look more familiar and maybe not as familiar for me, which industry is pursuing. So in basically very short period of time, I can get a really good picture of what's happening in the industry and also be able to just with a click of a button get to the patent of interest really saves a lot of time. And maybe if I can, I would also say that this goes true also for the molecules of the month flyers, which drug hunter teams really distills, taking pretty much all of the medicinal chemistry literature for us and looking through the flyer and just picking a molecule which captures my attention and then being able to very quickly go and read about it is also a very nice feature. So I really appreciate all the work Drug Hunter team is doing for us, really distilling science into very very amendable to to pursue vignettes. So
Awesome. Yeah. Thank you so much, Alexi. Really appreciate it. Yeah. Before we get started with your presentation, I want to show maybe one quick feature that we just introduced this week on Drug Hunter, a new feature that we launched. So, sharing my screen here. Um, you can find the feature here. It's a calculator. If you go to resources and go to the new scaled clearance calculator, just click on that one. And what you can do here is you can basically put in your, let's say, liver microsome clearance from any species. You can select your species here. So let's go with mouse. You can select the assay. You can put in either microsome or hepatocyte data. And then you can or cannot put plasma protein binding. If you don't, it's basically adjusted to 100%. But let's put a number, And then you will get your scaled clearance here, as well as your hepatic clearance. In this case, from a microsome assay with mouse, you can kind of change through the species and see how that number kind of changes, as well as change through the assay as well, putting microsome data as well as hepatocyte data here as well. So we really, personally, I was kind of using tables when I was in medicinal chemistry. So we thought it would be easier to build a calculator and hope that this is very useful for you. Please let us know if you have any feedback and if that's useful for you. And with that, I will pass it on to you, Aleksey. And we're very excited for your presentation today.
Thank you, Dennis. Let me just share my screen. Is it coming through?
Yes, perfect.
Okay, great. All right. Thank you very much, Dennis, and thank you the whole Drug Hunter team for this opportunity to present today. So what I want to talk about today is how physics-based modeling can be helpful for medicinal chemists to guide their decision-making on the drug discovery campaigns. I will first introduce the physics-based modeling, and we'll very briefly talk about some of the tools we have in our digital chemistry platform. I will then take you through three drug discovery stories from the programs which we worked within Schrödinger Therapeutics Group. And I will focus specifically on how FEP can actually be used to optimize both potency as well as the selectivity. I will also want to spend some time talking about the QM method, which we recently developed for brain penetration and efflux optimization. Schrödinger, as many of you might know, have a 30-year history developing the digital chemistry platform, which is very broadly used by pharmaceutical, biotech industry, as well as by academic institutions. So our digital chemistry platform integrates both physics-based as well as the AI and machine learning methods, and also covers all of the stages of drug discovery, from essentially target validation and enablement to the preclinical compound nomination. We developed methods which help with the protein structure refinement as well as the druggability assessment, and have a whole suite of tools which allow for large-scale virtual hit finding. What I will talk about today is how we use the in silico MPO built on the properties predicted by accurate computational methods during the drug discovery programs to essentially minimize the number of compounds which needs to be synthesized to achieve the desired target product profile. We've also been developing the tools to help with preclinical development. With this, let me introduce FEP plus, or free energy perturbation methodology, which some of you on the call might be familiar with. This is the computational assay, which allows for the accurate prediction of potency and selectivity. One of the typical questions which medicinal chemists face is on the program upon getting experimental data for their compounds and generating the new round of designs, whether this newly designed compounds will maintain potency on the target or will be more or less potent. FEP allows to answer this question by predictive relative binding affinities between the new designs and the reference molecules. And during the simulation, the structure of the reference compound is perturbed alchemically to the structure of the ligand of interest in both solvent as well as the ligand protein complex. These high accuracy simulations take different contributions to binding into the account, both enthalpic, entropic, water energetics, as well as the flexibility of ligand as well as the protein. The output of this method is the difference in free energy of binding between the reference and the new design, which can then be converted to predicted KD, Ki, or IC50 value, depending on the data available for the reference compound. In case of the optimization of potency, so free energy perturbation simulation can be run on the protein, on the off-target protein, with the goal now to identify the compounds with predicted low potency and therefore showing the selectivity against that particular off-target. And I will show you real-world examples of this later in the talk. So we often refer to the FEP as the computational assay because the accuracy of this method is now approaching experimental levels for the amendable targets. The graph on the right is just a representative example of what correlation between FEP predicted potency versus the experimental affinity can look like on the real program. So each of the blue dots on this plot represents an individual compound. And as you can imagine, if all of these blue dots would have lined up on this black unity line, that would have represented the perfect correlation between prediction and the experiment. Of course, in the reality, there is some variability. There is also some outliers or exceptions because none of the methods are absolutely perfect. But what's really important, that majority of the compounds essentially line up in this dark gray shaded area, showing very good correlation between predictions and experiment. And this level of accuracy makes this method very powerful for us to select compounds for synthesis of most promising analogs. As just an example, in this particular case, if medicinal chemists would be making a decision based on the FEP predicted values of PIC50 of more than 8, then the large number of inactive compounds will actually be deprioritized during the selection process, and they will never be made experimentally. On the other side, there will be a good enrichment on the compounds showing high experimental potency, including identification of these two most promising analogs on this particular program. What's also really important is that we now can run this method at a much faster speed comparing to what takes us on average to make the compound in the lab. And with the current computational power, we can also have a fairly high throughput, profiling a large number of compounds through this method. Of course, potency and selectivity are the only two important endpoints which we optimize during the drug discovery campaign. So therefore, we incorporate other important compound metrics which are predicted through the computational methods into this in silico multi-parameter optimization, which essentially is at the heart of what we call DPMTA cycle, where P stands for predict. So incorporation of this predictive modeling prior to the design prioritization for synthesis makes it really powerful to prioritize compound based on their predictions rather than ease of synthesis, and only pursue the compounds which looks exciting from the predictive standpoint to test a particular hypothesis. So this predict first paradigm essentially encourages us to explore a pretty large chemical space, and in the end leads to the reduced number of the DPMTA cycles needed and number of compounds needed to be synthesized to reach the exciting target product profile. So with this, let me shift gears and now talk about the three drug discovery programs which we worked on within the Schrödinger Therapeutics group, representing diverse targets as well as the protein families. I will start with our wholly owned MALT1 program, where we were able to identify the developmental candidate SGR-1505 in just 10 months since the beginning of the program. So the goal of this particular program was to design novel, potent, selective allosteric MALT1 inhibitor. And we started this program from compound 1, known in the literature, as well as its co-ligand, the structure available in the public space. In our initial designs, we focused on the two parts of the molecule, core 1, as well as the linker, keeping this core 2 part of the molecule constant. So if you look through our screening funnel, we designed a total of about 1,400 compounds, and vast majority of the compounds went through docking and filtering, and we prioritized for free energy perturbations to predict their potency. Only small percent, or about 3% of the compounds, 37, was then prioritized for synthesis based on the category A and B predictions. In FEP, we were essentially looking for the compounds which were predicted to have a potency of less than 100 nanomolar or comparable to the original compound. What was also very interesting and surprising for us to see that for the subset of compounds where we were removing one of the hydrogen bond donors from this urea substituent that had an interaction with their protein, the potency of this compound was predicted to be comparable to the reference. So it was really exciting and non-obvious for us, and we were really curious to test this hypothesis experimentally. So from our very first DPMTA cycle, we have discovered multiple novel hit series, and one which was this compound 4 with a novel tricyclic core, which also showed comparable potency on target comparing to the reference. So other chemical series were either less potent, also not chemically stable, but was very interesting to see their experimental affinity aligned pretty well with the category B FEP predictions. We also generated the co-crystal structure of compound 4, showing that indeed only one of these key important interactions is needed to the glue 397 for maintaining potency, exactly as was predicted by FEP. So this compound 4 was a really great starting point for us. So it had good biochemical as well as the cellular potency. It had good properties, good mouse and liver microsome stability, as well as the oral bioavailability. It was also the first compound which demonstrated dose-dependent PK/PD, as well as the tumor growth inhibition in the CDX model. So the next important step for the program was to further improve on-target potency. And one of the directions we undertaken on this program was exploring the small hydrophobic pocket next to this gem dimethyl substitution. We explored about 300 ideas by FEP and progressed only a small subset of ligands into the synthesis, which were predicted to be interesting from the potency perspective. Among those compounds was this compound 7 with the stereospecific trifluoromethyl substitution on the score, which was predicted to be single-digit nanomolar potency on target. I also would like to highlight that accurate FEP prediction often encourage medicinal and synthetic chemists to pursue challenging high-quality ideas such as compound 7. In this case, it actually took the chemists 4 months and pursuing 11 different synthetic routes to introduce the trifluoromethyl group at this quaternary center. But all of that effort was very well worth it, since compound 7 was indeed tested at 1.2 nanomolar in the biochemical MALT1 assay in good correlation with the FEP predictions. Excitingly, this biochemical potency nicely translated into the cellular potency, and this compound also retained good properties and was a key analog for the program to transition into lead optimization. In the interest of time, I won't take you through whole lead optimization effort, where we continue to explore a broad chemical space coupled with a multi-parameter optimization to further optimize compound 7, which I just talked on the previous slide. Excitingly, in just 4 months since we started the lead optimization program, we identified SGR-1505 as our developmental candidate for this program. It was only 10 months since we started this program. We also have made a fairly small number of compounds, and in the leading series, we only made close to 80 analogs to identify our developmental candidate. So this compound is currently in the clinical trials in patients, and we're really excited to report some preliminary clinical data later in this year for this compound. So the second story I would like to talk about is the collaborative work we did with Morphic Therapeutic before Morphic became an independent subsidiary of Eli Lilly through the recent acquisition. So Morphic was founded to discover orally bioavailable small molecule drugs for various integrin targets. We built a truly synergistic and collaborative relationship between teams from 2 companies, where Schrödinger team was involved in design and medicinal chemistry activities, and also was responsible for driving all of the modeling activities for the program using the proprietary crystal structures from Morphic, which were absolutely essential for us to enable modeling on this complicated heterodimeric proteins. So Morphic team was also executing all of the experimental activities on this program as compounds were progressing from the design cycles. So the goal of this particular program was to discover first-in-class, highly selective alpha V beta 6 orally bioavailable antagonist. So when we started the program, Morphic had a small proprietary library, from which we were able to identify early hits, as well as to generate some early SAR to guide our future efforts. So compound 1 was tested at submicromolar potency against alpha V beta 6 in the biochemical assay and showed fairly limited selectivity against closely related integrin alpha V beta 1, which differed in this beta subunit. So these compounds bind to the RGD pocket of this integrins with 2 key anchor interactions in 2 subunits. One is the carboxylate, which interacts with the magnesium atom in the amide site, and then the THN was anchored to the aspartate 218 through the salt bridge. The rest of the molecule is essentially serves as a linker spanning across the interface between alpha and the beta subunit. So the first important modification which we showed is the incorporation of this alpha phenyl group, essentially pointing toward two of these selectivity determining loops, SDL1 and SDL2. This had a very pronounced impact on potency, as well as for the first time we saw moderate levels of selectivity against this closely related off-target. The challenging part with these amide linkers, whether they were linear or constrained, that none of the compounds actually had good permeability. That really precluded us from ability to develop orally available adjunct. Through additional SAR, we were able to show that incorporation of this flexible ether linker led to the significant improvement in permeability. And we were really interested to understanding why. So obviously we are removing the secondary amide from the structure, but what was actually more important in this case is the ability of the molecule to form this shielded conformation by intramolecular hydrogen bond interaction between two charged sites. So we essentially masking carboxylate and THN through this intermolecular hydrogen bond interaction, which we were able to model by using the permeability modeling. So if we would remove one or two atoms from this linker, this compounds actually lost significant amount of permeability, again, likely due to the inability to form this intramolecular folded conformation. So the next important step on this program was optimization of both potency and selectivity as we settled on this pyrrolidine ether scaffold as our lead series. So looking at the co-crystal structure of the compound 3, it was evident that growing toward this SDL2 loop could lead to the improved potency. We explored both of the possible vectors, ortho as well as the meta, and very quickly settled that ortho substitution was more productive for gaining potency. So as you can see from these three examples, both types of groups, small ring sizes as well as the linear substituent, were very well tolerated for the experimental potency, gaining about tenfold comparing to the original compound, in good agreement with the predicted numbers. So in order for us to find the additional 10 boost of potency, which was needed to drive the on-target alpha V beta 6 potency to single-digit nanomolar level, we needed to explore really large number of compounds, both through hand-drawn designs as well as through enumerations, and we profiled them computationally, looking for the compounds with the best predicted FEP potency. So one of the productive and exciting exercises we have done was looking at the subset of this congeneric THF and THP substituents, which were connected to the phenyl group through different positions with the different stereochemistries. One could ask a question, which of those ligands would have the best predicted potency? And I could just say that none of these compounds had any specific interactions with the protein. It was really impossible to predict this looking at the docked poses. This is where FEP was very handy. And out of all of these possible isomers, FEP had the preference for one specific S isomer of the THP, predicting single-digit nanomolar potency on target, whereas the rest of the compounds showed a fairly flat SAR with predicted PIC50 hovering around 8. So really inspired by this prediction, the team didn't shy away from figuring out chemistry and making compound 7, which indeed was tested at 1.6 nanomolar in alpha V beta 6 assay in a good prediction with our FEP prediction, and also provided us with this needed potency breakthrough on the program. So in addition, this compound now showed moderate selectivity against two closely related off-targets I mentioned earlier, alpha V beta 1 and alpha V beta 8. So we also generated the co-crystal structure of this compound in alpha V beta 6, confirming the absolute stereochemistry of the center, as well as the lack of any specific interactions with the protein backbone. So in the next stage of the program, we set a goal to improve selectivity against the key off-targets to more than 100-fold, essentially trying to avoid any significant coverage, even at the Cmax for these targets. So looking at the co-crystal structure, which were very, very helpful for us in the design, in guiding our design methods, we saw that there is an additional space for us to explore under the selectivity determining loop. So starting from compound 7, we did the systematic gem dimethyl walk around this THP ring, and then profiled all of these regioisomeric compounds in 3 FEP models. We were first looking in alpha V beta 6 FEP for the predicted potency, whether they were maintaining potency on target, or we saw the improvement. And then the compounds we also profiled in alpha V beta 1 as well as alpha V beta 8 receptors to assess their selectivity. As you can see from this table, 2 specific isomers with substitutions at these positions emerged as the most promising analogs, showing improved potency, as more importantly, improved selectivity against off-targets. So we went ahead and we synthesized these most promising analogs first, and we were really excited to find out that compound 8 showed the sub-nanomolar potency, essentially now being at the bottom of the biochemical assay, and very importantly, had a significantly improved profile comparing to the reference analog, consistent again with FEP predictions. Out of the scientific curiosity, we have also made another analog which was not predicted to have any advantage over the original non-substituted reference, and it was also confirmed experimentally. We never pursued the analog which had lower predicted potency and selectivity, since it wasn't in the program's interest at this time. Through further quick optimization of compound 8, through incorporation of this important fluoroatom to improve the permeability and oral bioavailability, I don't have time to talk about the whole story behind modulating the PK of this particular center, but that's what this fluorosubstituent was doing for us. And additionally, we cyclized the gem dimethyl group into fused cyclopropyl to arrive to compound morph 627, which became the developmental candidate for the program. So the last drug discovery story I want to talk today is from DLK program, where we had a goal to discover best-in-class orally available and CNS penetrant small molecule DLK inhibitor. In our hit finding strategy, we started with a known hinge binder from the work reported by Genentech, and took a stepwise approach to first identify novel cores and then decorate them with different R groups, an approach which was guided by FEP. During the course of the program, we needed to further optimize the core of the molecule, as well as do some modifications to the hinge to get to the optimal compound profile. So this slide highlights the large-scale FEP process to identify the novel cores. Similarly to what I described for the MALT1 program, we enumerated 2,400 ideas using the library of monocyclic and bicyclic cores, and the vast majority of this idea survived filtering and docking part, and 85% essentially progressed into the single-edge FEP. This is the fast version of FEP, which allows us to quickly prioritize the most promising compounds, saving on the computational cost. As you can see, only a small subset of compounds then progressed into the full cycle closure FEP to get accurate predictions. This is a good illustration of how FEP can help very quickly to narrow down the list of compounds which we can consider for synthesis. So small subset of compounds with synthetic feasibility then progressed into the synthesis, and in just 2 months since we started this exercise, we were able to generate a number of novel ligand-efficient DLK hits, which are shown in this box. One particular series which was really attractive for us was this imidazole pyridine, based on their experimental potency as well as the ligand efficiency, and also some of the early ADME DMPK data emerging for this series. So I won't have time to talk about the whole medicinal chemistry campaign in this program, but I will just highlight a few key steps on our route to identify pyrazole-pyridinone core, which became the key series for this particular program. So we started from this imidazole pyridine core and was very quickly able to identify the substitution patterns, which allowed us to drive DLK potency to sub-nanomolar level. I again want to show what the typical distribution of potency looks like during the FEP simulations, comparing the compounds which have various R substitution patterns to the unsubstituted analogs shown in this red dot. As you can see, the FEP method allows us to focus on most promising ideas experimentally, and with only 16 compounds synthesized in this particular series, we were able to get to compound Chi 3723 with this sub-nanomolar potency on DLK. Unfortunately, this compound suffered from very strong hERG inhibition, and we needed to do additional tweaks to the structure of the molecule. What we have done later in the program, we replaced this electron-rich imidase pyridine core with the pyrazole pyridinone, did an additional tweaking to the hinge binding motif, and also replaced this basic amine with a neutral THP substituent. These modifications allowed us to maintain excellent potency on target, as well as achieving brain penetration, while addressing the hERG and CYP340DI liabilities, which we observed for the earlier analogs in this series. One key important challenge which was left for us in the program is addressing the kinome selectivity, which was actually pretty suboptimal for the compound Chi-10634, in particular for the program which was targeting neurodegeneration. During the lead optimization stage of the program, we established a very efficient workflow, which included a number of physics-based methods, both QM and FEP-based, to prioritize compounds for synthesis through this in silico multi-parameter optimization. So the two specific tools I'd like to talk in more details is use of this ESOL modeling to optimize brain penetration or KPUU, as well as the FEP modeling to optimize kinome selectivity. I invite everyone to read through our recent manuscript, which came out in JMedChem for the full medicinal chemistry story, including use of the other predictive modeling during the course of the program. By profiling this compound, Chi-10634, through the ScanMax panel, we were able to identify a handful of off-target kinases, which are shown in this table, along with their fold selectivity windows relative to DLK. Also, based on the analysis sequence identity analysis, we showed that we could use potentially FLT3 as the surrogate for optimization against other two off-target kinases, TRK and KIT. So computational chemists very quickly were able to establish FEP models for all of these four kinase off targets. And the performance of the FEP method was really good, showing good correlation between the predictions and experiments, very well aligned in expectation for the accuracy of this method. So this level of performance gave us confidence to now use what we established as FEP selectivity score, which was calculated based on this FEP predictions in each individual off-target kinase. So at this stage of the program, we ran all of the compounds through 5 FEP models, first in DLK to make sure that the compounds retained on-target activity, and then through 4 off-target FEP simulations to generate this FEP selectivity score, which we also showed early on that has a good correlation to the kinome experimental selectivity. Using this FEP off-target workflow, we were able to improve the kinome selectivity very quickly, starting from compound 10634. So very interestingly, early on, we were able to show that introduction of this methoxy groups in the cis relationship on the THP group led to the predicted improvement in the selectivity, which nicely correlated to improved selectivity experimentally, which we observed for this compound Chi-10788. Then further, through the couple additional DPMTA cycles, we were able to identify compound Chi-11101, which had the highest predicted FEP selectivity score of 0.98, just to compare in drastic difference comparing to the starting compound 10634. We also confirmed that it matched our experimentally observed MPO really nicely. And what was really important for us is that overall kinome selectivity profile, as shown by the ScanMax experiment, significantly improved comparing to our starting point. So let me finish my talk by talking about using the ESOL modeling for optimization of the CNS penetration for this DLK inhibitors, which was one of the key challenges on this particular program. So early in the program, as we were optimizing the compounds, we showed that the non-commonly used CNS prediction metrics can have a good correlation to the experimentally measured KPUU. That included the use of the CNS MPO scores by Pfizer or Merck, as well as some other parameters such as log-D or PSA. We showed that there is some enrichment can be achieved by using the MDCK MDR1 efflux data for this correlation. But what was really exciting for us is that our computational team at Schrödinger have recently developed a QEM-based method of calculating ESOL, or energy of salvation, which showed really impressive enrichment and correlation of brain KPUU to the ESOL using the rodent brain PK. So this energy of salvation is the QM calculated property of the molecule, which is essentially the partition between water and the vacuum using the implicit water model. So this particular calculation factors in different, takes into account different contributors such as 3D molecular conformation, size, partition, polarity, as well as the energy of neutralization, which is essentially the pKa. You can think about it as a physics-based metric, which essentially replaces the empiric MPO. Using this method, we demonstrated really good correlation between KPUU values to the ESOL for the DLK inhibitors from various chemical series. For the compounds which had higher negative predicted calculated values than -18, typically had very low probability of achieving a target KPUU of more than 0.3, whereas for the compounds which had ESOL values calculated of more than -16, typically had highest probability of achieving this desired KPUU. There was also a gray area for the compounds between -16 and -18 kilocalories per mole. So we then started to utilize these ESOL calculations in our in silico multi-parameter optimization to really help us prioritize the compounds with a higher probability of achieving high brain penetration. And let me now show you several of the specific examples or non-obvious examples which we came across during this program. So it includes matched molecular pairs from both basic containing amines on the solvent side, as well as the neutral THP analogs. So the question which one could ask, which of these compounds will have the best experimental KPUU? And depending on which particular CNS metric you would use to trying to answer the question, you'll probably come to a different conclusion. As an example, it would be basically impossible to make the predictions based on the MDCK MDR1 efflux, as all of these compounds had efflux ratio of less than 2. Here is the experimental data, which showed that the compound with the more basic unsubstituted piperidine had much better KPUU comparing to the compound with attenuated pKa with this oxetane substitution. Also, very interestingly, this ethoxy substituted THP analog had much better KPUU comparing to the difluoroethoxy compound and comparable to the unsubstituted tetrahydropyran, which while still having the polar functionality. So what was really exciting for us is to see that there was a really good, this can be explained by ESOL calculations, where the highest negative ESOL values was observed for the compound Chi-10-606, as well as Chi-11-048, consistent with their poor brain penetration. So interestingly, we also continued to look for the ESOL correlation to KPUU across different CNS projects within Schrödinger, as well as for the published data sets. And we were able to show that the 77% categorical accuracy could be achieved for 300 compounds targeting KPUU of 0.3 using, again, ESOL cutoff in this case is -18 kilocalories per mole. So I'll just like to highlight that the categorical accuracy in this case is calculated for ability of the method to place compounds accurately into the true positive, true negative, false positive, or false negative quadrants. We also were able to show that the ESOL calculation can enable MDCK MDR1 flux optimization. So in this case, we actually targeting the efflux ratio of 2, and we were able to show that 80% categorical accuracy could be achieved to help us with the efflux optimization. In this case, we use the ESOL values of -19 as the cutoff. I think I'm slowly get running out of time, so I'll just briefly mention that we can also use these methods to enable the Keiko-2 efflux optimization. In this particular case, it's a little bit noisy, but we can still achieve pretty high levels of accuracy, targeting efflux ratio of 5 to maximize the compound's probability of achieving oral bioavailability. And just to give you a quick flavor, here is a very interesting match molecular pair where two compounds published recently in JMedChem publication had very different Keiko-2 efflux profile, and we were able to explain it with ESOL calculation showing significantly higher negative value for the compound 27 comparing to compound 28, and this method was actually able to put these compounds in the corresponding quadrants accurately. Again, this is the physics-based method, which allows us to optimize efflux for both Keiko-2 as well as MDCK MDR1. So this brings me to the end of my talk, and I would just like to thank everyone who contributed to the programs I was able to tell you about today. And thank you all very much for your attention. I will be very happy to answer any questions.
Awesome. Thank you so much, Aleksey. That's super impressive. I'm a big fan of this ESOL method. Like you just showed basically that it can differentiate between two diastereomers effectively, right? And predict that the efflux ratio. So really, really surprising. Cool. So we have a lot of questions from the audience. So it speaks how phenomenal the talk was. We have more than 20 questions. I'm not sure if we can get through all of them, but I'll try to sort through them a little bit. So you showed this nice funnel where you kind of started with, the selection of some compounds, then did docking, and then, whittled down, I guess, to FEP, right? FEP plus. How much human in the loop input is there from experts, medicinal chemists, is what Ash is wondering, and is some other people in the audience as well.
Yeah, so medicinal chemists play a very important role in the, in selection of the compounds for synthesis. So they basically define what property space we want the compounds to be. So even at the very top of the funnel where the filtering is happening on the property space, that's where the medicinal chemists define what that space should look like. When the number of the compounds becomes manageable, they start really looking with the computational chemists together, picking the compounds which make sense to pick for synthesis. So there's still a good amount of medicinal chemistry intuition, which is very helpful during the process of selection compounds through synthesis while triaging large compound libraries.
Sounds good. And that's the way you also select those compounds that you want to select for docking. So we had a question here in the MALT program. There were about 1,400 compounds selected for docking. It's by kind of collaboration between human input and computational chemists, right?
Yeah, so in that particular case, the 1,400 compounds which I showed, they were mainly generated from hand-drawn designs. So they all just progressed into the docking and filtering just to make sure that they fit the binding pocket. So in some other cases, we also use very large enumerations where the number of compounds is significantly larger. And then they actually do progress into the first steps kind of all together. And then we see the best compounds emerging from that first step of the funnel to then further prioritize for the next selection process.
Yeah, makes a lot of sense. And then for the DMPK data, which kind of data do you use to train those DMPK models, given that structure-based approaches are typically not very applicable to many of these parameters? And how do you curate it for quality is what Ash is wondering.
Yeah, so that's a great question. So for the ESOL calculations, there is no training which is required. Again, it's the calculations of the fundamental compound property, right? The partition between the vacuum and the solvent. For the other methods, when we use AI and machine learning methods for the optimization of the DMPK properties, we carefully curate the libraries or training sets. We put a lot of thinking into what exactly the training set should look like. In our experience, the local models which trained on the specific chemotypes typically give us better performance comparing to the very global models where we include all of the data from many different projects, many different chemotypes. So I hope that answers the question.
Yeah, thank you. And then coming back to FEP, so Gerald from the audience is wondering, he's saying that's very nice to see such quick progress. Is there any examples or any times where FEP cannot be used for optimization? And if so, what are those types of cases?
Yeah, so that's a really excellent question, right? So in order for us to efficiently use FEP on the program, there is several requirements. So we do need to have the structural information for the protein of interest, right? So it could come in the context of the crystal structure or cryo-EM structure, or more recently as the predicted structural model. So we do need to have that information. So we also need to have a good understanding of the binding mode of the compound in that specific protein to be able to do the simulations. So those are the important requirements to do the relative FEP calculations. We also, for what I showed in the talk, we also needed to have a reference, right? We need to have a reference to be able to predict the relative binding affinity of the ligands of interest. Interestingly, we have developed also the different flavor of FEP, which is called absolute binding affinity FEP, which doesn't require having the reference, and we can actually predict absolute binding affinity, but we do need to know the binding mode of the compound. So those are the several requirements for being able to use FEP methodology for on the programs. But I think one thing I would say, definitely with the expansion of the cryo-EM methods and the number of structures which are now available to us, there is more and more targets which are amendable for using this methodology efficiently on the program.
Sounds good. Is there any examples, Irv is wondering, where FEP predictions have been misleading retrospectively, or
Yeah, that's another excellent question. As I mentioned briefly, right, none of the methods are perfect. So there are definitely cases where the FEP predictions would not line up with experimental data, but this is typically not a reason to give up. This is just an opportunity to go ahead and try to understand what was maybe different in the method and try to improve it to essentially get to the appropriate level of correlation between the experiment and FEP predictions to drive the program. So that definitely happens. Sometimes, as I said, you get the outliers of compounds which are predicted differently than what you see experimentally. But as we have shown on programs all over again, that if you continue working and optimizing the method, you usually can get to the very good levels of performance, making a significant impact on the program progression.
Awesome. Yeah, thank you. We have kind of an anonymous question. How has AlphaFold changed the workflow in the space?
Yeah, so that's another good question. As I just mentioned, right, we do need to have structural information for being able to use these methods. And one way to get structural information is from this predictive structure folding methods, such as an AlphaFold. And there are examples where we could use the structure with appropriate validation to be able to apply FEP methodology. So I'm definitely seeing in the future that this specific methods will make more and more impact on how many targets we can tackle with this methodology.
Yeah, makes total sense. And then for the Morphic program, Yoshi is wondering, you mentioned that it's hard to know which substituents to use, but FEP identified the THP ring. Were other methods, for example, Glide, not able to identify this THP ring?
Yeah, so as I mentioned, that pocket is actually pretty big, and it's a lot of different substituents can fit nicely under that selectivity determining loop in the beta subunit. So using docking or docking scores was not actually possible to really differentiate the compounds, in particular when they are so similar in the structure as well as in the substitution pattern. So FEP was really helpful for that program to identify specific compounds which led to the predicted improvement in potency, and then nicely translated into the experiment. So yeah, docking was not really possible to do those types of accurate predictions in that program.
Makes sense. Yeah, it's a nice differentiator there for sure. For the DLK program, or in general for the ESOL predictions, Ash is wondering, are they using a single low energy confirmation or multiple confirmations? If they use multiple confirmations, what's the computational cost, considering that it's a QM technique?
Yeah, so that's a good question. So this method considers all of the possible 3D conformation to identify the lowest energy conformation, and that's how ESOL value is generated. So one thing I also would like to mention, that this property can also be calculated not only by the QM method, but also as the part of the FEP calculation in the solvent. In that case, we call it as a hydration free energy. And that technique also allows very efficiently sampling a vast majority of the 3D conformations to essentially being able to predict that ESOL value accurately. So in terms of the computational cost, these calculations can actually be done in a fairly short order, just of a few hours. So they are very much amendable for large throughput.
Amazing. And then another audience member is wondering, have you tried the ESOL prediction for commercially available CNS penetrant drugs and kind of validated the model on those, maybe?
Yeah, so as I showed in one of the graphs in the presentation, that in addition to showing good correlation of KPU on the programs we had at Schrödinger, we also included publicly available data sets for the approved drugs or any other compounds we found in the literature with KPU values, and showed very good correlation. Again, showing the ability of this method to help us with optimization for brain penetration using the ESOL calculations.
Nice. And in that program, you also showed how you dialed out a Herc liability. Some audience members are wondering, what was the most important step? Like, what kind of method did you use to dial out Herc activity, and what was ultimately the most important change?
Yeah, so that's a great question. So as I showed for one of those key compounds we identified in the program, it contained a really highly basic amine. So we showed that by modulating the basicity of that amine, we can actually attenuate Herc liability quite quickly. So it's again using this physics-based QM calculation of pKa value accurately, which allowed us to really quickly address the Herc liability, even without removing that basic functionality altogether. Eventually, in our most advanced compound, we had a neutral substituent which solved the Herc liability altogether. But even before we got to the THP substituent, we were able to modulate it by attenuating pKa with accurate QM predictions.
Awesome. Yeah, thank you. And we're quickly coming up on time. Thank you so much for answering all these questions. Maybe one last question, Aleksey. And we still have 20 questions in the chat. Just
Happy to answer them offline.
thank you so much. Yeah, so last question is also from Ash. What is your process for designing commercially available building blocks during your modeling to synthesis and availability, especially for rapid early lead generation?
Yeah, so we have a curated libraries of various commercially available building blocks, which we can specifically use during our enumeration process to make sure that the compounds are actually accessible synthetically. So that's an approach we would use if we want to make sure that we early in the program generate compounds which we can then quickly progress in synthesis and essentially confirm the predictions experimentally. So we do use the curated libraries, which we keep track of for this enumeration purposes.
Yeah, sounds great. Well, thank you again, Aleksey, for an amazing talk. Really enjoyed all the 3 examples that you showed. I think you really showed us how can FEP be used, how can the ESOL model be used to predict brain penetration. I really enjoyed it, and I think so did the audience. So again, a big applause from our end virtually here for a very nice presentation. For all the audience members, thank you so much for your high engagement, for all the questions you submitted. Really enjoyed this highly interactive seminar. And we will be back next week with Nikki Goodwin and Neil Fazakerly here, talking about inhaled JAK inhibitors, where we talk about some lessons that were learned for lung delivery, as well as for how to address AO metabolism. So we will see you then. And until then, wishing everybody a nice weekend. And thank you again, Aleksey.
Thank you so much. It was really a great pleasure to give a talk.
Thank you. Bye bye.